Hello everyone, day 2 of the csRNA-seq course. Yeah, I thought to do a quick summary again. First of all, it was really cool to see how your awareness for working with RNA has increased. There were attempts to go inside bags of tubes and like, oh no, I shouldn't do that. Or occasionally tube opening. So that was really cool that you really take it to heart, and we have to remember the amount of RNA that we're now working with is so little. We couldn't quantify it with a qubit, and we also didn't see it in our gel. But then of course habits too. For example, we love opening tubes with one hand, and the way, for example, I broke this habit is that I only open tubes now when they are standing in a rack, and then basically I have to use one hand to open the tube while it's in the rack. And as I never did that before, that was a new way of opening tubes, and that's how I got around my habit, that although I still use it when I'm cloning, to open the tube with my thumb. Two things, or a couple of things from today that I wanted to emphasize one more time. The first is the salting out. You all knew it and did it well, but we have to remember we first have to add the salt to the water, mix it before we add the ethanol or the isopropanol. This is so that the RNA is then highly polar, while the ethanol or the isopropanol is unpolar. And it's actually the repulsion between polar and unpolar that makes our RNA fall out. So if we don't add the salt in the RNA and pre-mix it prior to adding the isopropanol, the RNA doesn't, the salt doesn't mind mixing with the isopropanol, but then it doesn't sort of get chelated by the hydroxy groups of the RNA and the phosphate backbones quite as well. Another comment was about Glycoblue. A couple of you had sort of various intensities of pellets. Glycoblue, I should emphasize again, is a very high molecular weight molecule. So if we defrost it and then we spin it really hard, the bottom will have more Glycoblue as the top. So it's one of those buffers that we want to vortex, and if we quick spin only very briefly before we use it. Another fun thing is, why do we use 60 microliter of sodium acetate? It's because I expect you to get about 540 microliters out of it. So when we talk about 1/10, that means it's 10%. So it's a 1:9 ratio. It's not as if I expect you to get 600 microliters out of this, because 600 plus 60 would be 660, and then it would not be 300 millimolar total. Another question was why there is IAA in chloroform. You should always have IAA present when you work with RNA. This is because it protects the RNA from degradation by chloroform. Okay, and looking at the protocol, then there's one more thing that changed between the protocol and what we did today. So I want to emphasize that one more time, which is we did not use the CYP2 master mix. And the lucky thing is, what I was just able to check before the class started, Quicksip was just released by NEB. It's basically a phage display optimized version of CYP. And so before we used it, I tested it, and Quicksip works like CYP, so that's good news. But the other great news about Quicksip, it is actually thermostable. In this sense, we do not need to add new CYP, because during the heating and the cooling down, we lose some activity of the CYP, but not all of it. So why do we heat it up and cool it down? Yeah, I mean, most of you know already, it's because the RNA has secondary structure, and those phosphates or unwanted 5' ends, or actually only phosphates because it's CYP, that the CYP should dephosphorylate because it's a phosphatase, right? They might have been covered in the first round, and through the heat denaturation are now revealed. Okay, then tomorrow we're going to start again with a 30-minute spin. So I really like how you guys are all helping each other out, and I think we should build on that. So if some of us are running late tomorrow because of kids, daycare, and life itself, just reach out to your friends, let them start the spin. The first part of tomorrow will be exactly the same as today. We spin down the samples, we wash it with 75% ethanol. With one small point is, the moment we add the 75% ethanol, then we use the P1000 to transfer the pellet into PCR strip tubes. I would recommend that these PCR strip tubes have the same order as your input samples that you use today. For tomorrow, you will also see that there was a magic fairy that visited your boxes, at least those of you who behaved well today, and I'm not sure which other stories your parents told you, but anyway, you will find T4RNA ligase buffer and PEG in your boxes, which is what you require for the first step. I want to briefly comment again on working with PEG. PEG is highly viscous, so I recommend using the wild boar, the white orphanage tips tomorrow, and pipette it really slowly. It's a bit of pain of working with PEG, but it's also really cool because it strengthens your hand muscles, because we really want to shake the hell out of those samples tomorrow. Then one more thing is that I would like to start collecting a list from each of you about your samples and their names. This is because I would like to use the couple of breaks tomorrow, and maybe I should comment on that. Tomorrow is by far our busiest day, but the advantage is that the busiest people of the day will be our enzymes people. So we're going to have a bunch of incubations, but when we're smart and always use the breaks to help each other out, we have 2 aliquots of all the reagents. So if we pass that through, maybe group 1 and 2 going to pair up, and group 3 and 4 going to pair up and pass the reagents, we can always make sure that we have our master mixes ready and in the lids of the PCR strip tubes by the time the first incubation is ready, so we just have to exchange lids. By that, the day should be fairly smooth, and I'm pretty sure those who stay focused on the task will be out of here at 3:30 at the latest. But in these breaks, it would be also nice if you write me a list of all the samples that you have. And this is because I need to assign barcodes to you and have to start organizing in my head how we're going to sequence all of these samples so you get your results as soon as possible. For that, I draw this really funky diagram here. Oh, let's put it on there. Which basically says initial species cell type treatment. If you have several replicate, I would recommend you to use a -R1 dash and then csRNA-seq and then a unique identifier. The initial, I will just need to make sure that each of you gets the right samples. The species in general, it helps to have that in there in case you ever send these FASTQ data or you submit it to Geo, that people can just quickly analyze it and use the right genomes. The cell type in the treatment is really the information that allows us to know what you did. And then small r1, small r2, small r3, depending on how many replicates you have. This is really cool because if you have dash lower r1 dash, a lot of bioinformatics scripts automatically recognize that as replicates, which allow us to merge these samples, and it just makes the work with the samples easier. After that, here is the csRNA-seq, which is basically the method. And then I also suggest an identifier, and I maybe want to spend another couple of words talking about the identifier, which is, for example, in the glass lab, I used DG for Dutke glass, now I use DB, and I generated about 2,800 libraries by now. But if you would wake me up at 3 a.m. in the morning and you say, Sasha, I need 5 million more reads of this library, I can go to my Excel sheet, look at the DB number, know exactly what's in the tube, and I would also know where it is, which box, and so forth. So I highly recommend that to sort of have control over your samples even a lot later. Cool. With this, that's everything for today. Again, great work. Looking forward to seeing you all tomorrow. You will also get this information at least briefly written up via email. And one more thing for those of you who listen and now asking, how about csRNA-seq and input? Well, yes, if you look at that, I first want you to do all the csRNA-seq samples and then all the input samples. And this is just that because barcoding we will in partly do in 8 strips, and our csRNA-seq samples are on 1 strip and our input on the other. But then when we load it on the gel, we load csRNA-seq next to input. So yes, barcode 11 will be next to barcode 72, but that's fine. With that, have a good day and see you tomorrow. Bye.